One of the last notes that we left on in the bone tissue video was talking about how the trabeculae in spongy bone is arranged in such a way that the long end, the strong end of the plates and spicules in spongy bone line up with stress lines. Right? The more stress you put on an area, the bone will actually line up to make that area stronger. And that implies that bone, not just the organic portion, but also the inorganic portion, is dynamic and changing. And when you think about it, that makes sense, because bone has to come from somewhere. We know that bones grow over time, and we know that bones can heal. And so that means that bone tissue must be changing, must be malleable. And that's going to be our topic for this video. So the process of bone development, or at least the formation of bone tissue, is called ossification, or osteogenesis. Technically, osteogenesis means making bone, genesis, beginning, osteo, bone. Ossification means, so to ossify means to make like rock, or to mineralize, or anything like that. So technically, ossification is the process of actually laying down the inorganic hydroxyapatite that turns other sorts of tissue, cartilage or whatever, into bone. Our lecture here is going to be split into sort of two parts. First, we're going to talk about how bones are formed in infants, so how they're originally formed, and then we're going to talk about how they grow and develop over time in, children and, people going through puberty as their bones are growing, as well as how they change and remodel in adults. When we're talking about how bones are made, like how they first come to be in a developing fetus, there are two methods that are used: intramembranous ossification and endochondral ossification. And I'm going to go through each of these methods individually. So intramembranous ossification. Remember, ossification means developing bone, and here intramembranous means within a membrane. This is the way most flat bones, short bones, and irregular bones are going to develop in a fetus. So first off, you start off with, we got to talk a little bit about some embryonic development here. So when an embryo is first made, like you have a fertilized egg cell and it starts dividing, and it's dividing into other embryonic stem cells. One of the first divisions that gets made in the embryo is that your embryonic stem cells become one of three types of tissue. What's called the endoderm, the exoderm, and the mesoderm. And the ectoderm becomes like, your skin and your nervous system tissue, and your endoderm becomes a lot of your internal organ tissue, and the mesoderm, meso means middle, that's what will eventually become most of your connective tissues. So a mesenchyme, which you see here and all over the place. This is a tissue structure that basically mainly comes from mesodermal embryonic tissue, and it will develop into connective tissue, right? So when we talk about mesenchyme or mesenchymal cells, those are structures that you'll find in embryos and fetuses, not in adults, but it's like pre-connective tissue. It's the stuff that you have that will eventually become connective tissue. So we have our developing fetus, and we have some blood vessels that have already been made. They've got blood flowing through them. And we have mesenchyme or mesenchyme in between these blood vessels, and we have areas of osteoblasts. Remember, osteoblasts build bone, and specifically what they do is they lay down the bone organic portions of the extracellular matrix, and that is what they're doing. Everywhere here that is lined with these osteoblasts are busy laying down the collagen that will form the organic portion of bone tissue, and that's, you see here, this in blue, that says osteoid tissue. Osteoid tissue is bone tissue that has not yet calcified. So it's got the organic portions of the extracellular matrix, but it doesn't have the hydroxyapatite attached to it yet. So these osteoblasts start off by depositing the osteoid tissue, right, around wherever they're arranged. In step 2, we have that osteoid tissue has been made, and most of the mesenchyme has been turned into osteoid tissue, and the osteoid tissue begins to calcify, right? So we actually we attach calcium hydroxyapatite to the extracellular matrix, the collagen, and everything like that, and we get the formation of the first true bone. And so you can see that since this is forming around all of these blood vessels here, right, what we get is very thin plates of bone tissue, and these become your first trabeculae. So we calcify this osteoid tissue that was laid down by the osteoblasts, and when that osteoid tissue calcifies, it will trap the mesenchymal cells inside of it, and those mesenchymal cells will develop into osteocytes. So we have our osteocytes that are trapped in lacuna inside of our now mineralized or ossified bone trabeculae, the first bone trabeculae that was laid down. And we still have osteoid tissue because we're still growing our area. So we still have these areas of osteoblasts surrounding what are basically marrow spaces containing blood vessels and all sorts of other stuff. And they're still laying down osteoid tissue that will eventually be calcified. If we take a zoom out here in a greater overview, so we have all of this mesenchymal tissue has been ossified into trabeculae, and you still have these marrow spaces with osteoblasts coating them, and you still have this area outside where you have osteoblasts coating that as well, and we've formed our trabeculae. And basically, we've made the spongy bone here, and fibroblasts come by and lay down a periosteum, that dense irregular connective tissue, on the outside of our trabeculae, and then outside of that we have more mesenchymal tissue, which may be developing into some other type of connective tissue. The periosteum, right, has dense irregular connective tissue on the outside, and it has an osteogenic layer on the inside. So this very inner layer of the periosteum is osteogenic cells that turn into osteoblasts and then lay down basically osteoid tissue. And they just lay down a thick layer of osteoid tissue on the outside of this. So it starts off, step 3 here, we have a honeycomb of spongy bone with periosteum, and the osteogenic layer of the periosteum lays down a layer of compact bone. And we end up with, 2 thin areas of compact bone with our spongy bone in between. And the shape that this develops into is determined by the shape of these membranes that it's developing between. As I said, this develops the flat bones in a fetus and the clavicle, and in an adult, this is how your bones grow thicker. Not longer, thicker. As your bones thicken, this is the method that is used to do that. Here we can see a histology slide of this actually happening. So here we have osteoblasts, right there, right there. They're laying down. So these osteoblasts are actually captured inside osseous tissue. So this is mineralized. This here in the dark blue is mineralized tissue that has hydroxyapatite laid down on it. This area up here on the outside, this is osteoid tissue. So this layer here has the collagen laid down, but it doesn't have, it hasn't been mineralized yet. And then we have our osteogenic layer. This is where we'll have a bunch of osteoblasts and osteogenic cells that are depositing the osteoid tissue. And then out here we have the fibrous layer of the periosteum. And basically as this bone thickens, you just lay down more osteoid tissue. Osteoblasts lay down that tissue, and then they get calcified, and some of the osteoblasts get calcified inside, and they become osteocytes, and that pushes this layer, osteogenic layer, further out, and then you lay down more osteoid tissue. Not too difficult. Endochondral ossification, right? So remember how I said that bones come from cartilage? Well, this is the type of bone development where we have bones developing from cartilage, right? So with intramembranous ossification, we weren't actually making cartilage first. We were making osteoid tissue and then directly calcifying that into bone. For your long bones, they're going to develop through endochondral ossification. And in the early developing fetus, what you have is a hyaline cartilage kind of pre-bone thing, right? So you lay down this sort of vaguely bone-shaped hyaline cartilage. And you've got hyaline cartilage inside and a perichondrium surrounding it. The perichondrium is a layer of chondroblasts. Chondroblasts make cartilage. And and also an extra connective tissue layer. It's a dense, irregular connective tissue. It's very similar to the periosteum. It's just for making cartilage instead. So you start off by making your cartilage, hyaline cartilage sac. Right? Next step, our cartilage sac begins to develop into bone. First thing that happens is the perichondrium becomes a periosteum, and the chondroblasts are replaced with osteoblasts. And those osteoblasts begin secreting osteoid tissue, which then calcifies into compact bone tissue along the outside. So we're going to kind of ring it in bone, and that's called the bony collar. Second thing that happens is that some of these hyaline cartilage cells in here become what's called the primary ossification center. And they basically they die, and their lacuna are sort of replaced by osteoblasts, and the osteoblasts move in and start laying down bone tissue into this ossification center. While this happens, the chondrocytes inside the cartilage enlarge. They get bigger, and thus our pre-bone starts to elongate. In step 3, you have osteoclasts come in and burrow a hole through the bony collar, and you have blood vessels come in and nurture the primary ossification center. And in the primary ossification center, we're actually going to start carving out our marrow cavity. It actually, the tissue calcifies into basically bone first, and then osteoclasts come in and like chew away the bone, leaving sometimes some spongy bone behind. We have a blood vessel come in and begin nurturing this tissue. Our bone here in step 3 is still primarily made of cartilage, with a bony collar of compact bone around it. At this point, we have a secondary ossification center that begins in the epiphysis, in one of the epiphyses. At birth, so this is in a fetus. 4 is at birth. At birth, the diaphysis of the long bone has completely ossified. And so we have our layer of compact bone that was laid down by the periosteum on the outside. And the internal cartilage has been turned first into spongy bone, and then the spongy bone has been kind of worn away. Though when you're born, you actually have more spongy bone inside of your diaphysis. And the secondary ossification center has begun to undergo a very similar pattern, right? It's begun to calcify the cartilage matrix around it, and then osteoclasts come in and like hollow out that calcified area, creating a marrow cavity, basically a spongy marrow cavity, and we have a secondary blood vessel that's going to come in and start nurturing this area. And so we have a marrow cavity here, cartilage surrounding it, and then we have a metaphysis in here, and we'll talk about the metaphysis in just a second, but this is going to be a growing hyaline cartilage area. Down here at the bottom, we also have a metaphysis. We have a hyaline cartilage epiphysis that has begun to ossify with a secondary ossification center. In early childhood, early to mid-childhood, prepubescent childhood. What we have is, depending upon when you look, the epiphysis will have completely calcified into spongy bone on the inside. It will still have a layer of hyaline cartilage on the outside. That's what becomes your articular cartilage. And you still have a cartilaginous metaphysis in there. And in earlier childhood, then, an epiphysis might be present in cartilage form at one end, and usually at this point it's starting to ossify into a marrow cavity, and you have a metaphysis present. And then in adult bone, the metaphysis has grown, providing more area for the diaphysis, and the epiphyseal plate has fused, meaning that the metaphysis has grown and eventually calcified and become bone there. And you see the same on the bottom down here, with a single marrow cavity and closed epiphyseal plates. So the process of endochondral ossification doesn't end with birth. It continues up through puberty. So during infancy, like early, when you're born, basically, your epiphyses are made of cartilage, and they have an ossification center inside of them. At least usually the proximal end will have an ossification center inside of it. In early childhood, the epiphyses are made of cartilage for the most part. As you go through childhood into the beginning of your pubescent period, then the epiphyses gradually fill with spongy bone. Usually the way that happens is the cartilage will become calcified, right? So it becomes mineralized, but not in the very organized way that bone is mineralized. And then you have osteoclasts that come in and clear away the ossified cartilage and lay down trabecula the osteoclasts don't lay down trabecula. They clear away the ossified cartilage, and then osteoblasts come in and build trabeculae into that area, and so you end up with spongy bone on the inside, and you still have a periosteum on the outside that's laying down a thin layer of compact bone. Eventually, you're left with just a thin layer of cartilage at the end, and that becomes your articular cartilage, covering the joint surface. And post-pubescently, the metaphysis basically calcifies into the epiphyseal plate. So you have a metaphysis in early childhood, and it becomes an epiphyseal plate of cartilage, separating the primary and secondary marrow cavities, and that allows for bone growth and development. And that persists throughout childhood and adolescence until it eventually calcifies into the epiphyseal line at the end of your adolescent growth spurt. So here you can see an x-ray of a child's hand, and so this bone here, this is the diaphysis of the bone, and then you can see the epiphysis of the bone here and here. So there is non-bony tissue, cartilaginous tissue, in between the diaphysis and the epiphysis. You can see that it's a much more thin line on the proximal end and a thicker line on the distal end, and that's pretty common Those are the epiphyseal plates. So that's how bones develop in basically fetuses, in fetuses and young children who are still developing. Now let's talk about how bones grow throughout adolescence and into adulthood. And there's basically two ways the bones can grow. They can get longer, or they can get wider. And for the most part, flat bones and short bones and most irregular bones are just going to get wider. So they don't really have a long direction, so they just get wider, and then they hollow out more, and then they get wider, and then they hollow out more, and they just grow in all directions basically equally. Your long bones are going to grow in length, particularly in early childhood, and then again in your growth spurt in adolescence. And there's two different models for how this is happening. We're going to talk about elongation first. And bone elongation happening again mostly in the long bones. You have your epiphyseal plate. This is what the metaphysis becomes. This is a hyaline cartilage area between the diaphysis and the epiphysis. And this is going to be the growth zone where the bone will elongate from. This is called interstitial growth. Interstitial means from the inside. So we aren't laying down shells of bone on the outside. We're actually building bone from the inside, from this epiphyseal plate, which is going all the way through the bone. So here we have a metaphysis, or basically an epiphyseal plate. They're kind of interchangeable terms depending upon the age of the person you're looking at. But what we're looking at is, so here's the epiphysis up here, here's the diaphysis down here, and we are looking at this cartilage area in between and how it's going to get bigger. So what you see here, this is pretty much cartilage. And there is 5 zones in our growing metaphysis. Up here at the top is a thin area called the zone of reserve cartilage. This is not actively growing, but it is it's going to be what is attaching your bottom growing zone of the metaphysis to your top growing zone of the metaphysis. So this up here is the zone of reserve cartilage. Next, we have the zone of cell proliferation. Right? This is where you have your chondrocytes, your cartilaginous cells. They're dividing and making new cells with new lacuna. So if you take a look in here, in the zone of cell proliferation, you'll see that the lacuna, rather than being nice and large, will go into these flattened stacks. That's what happens as they divide. They divide, and then the lacuna splits, and then they divide, and then the lacuna splits, and so you end up with these stacks of flattened lacuna. You can see down here as well. This is where we're going to get more cells from. That's the zone of cell proliferation. Zone 3 is the zone of cell hypertrophy. We're still growing here, but we're growing a different way. Remember, there's two ways for tissues to grow. Tissues can grow by cell division, you get more cells, or they can grow by hypertrophy. Same number of cells, but the cells get bigger. So in our zone of cell hypertrophy, we cease mitosis, but the chondrocytes enlarge, and they thin the walls between the lacuna. So if we zoom in here, you'll see at the top of our zone of hypertrophy, we still have like these stacks of lacuna, but as you move further down into it, you see the lacuna are getting bigger, the cells are getting bigger, and the walls between the lacuna are getting thinner until we get down towards the bottom and we have no longer flat, but now big, round lacuna with fairly thin cartilaginous walls between them. That's bone growth through hypertrophy. In zone 4, we have the zone of calcification. In this area, what we're actually doing is not technically laying down bone, but we are calcifying the cartilage matrix. So there is a type of collagen in cartilage. It's not a very big type of cartilage or collagen, but it does exist there. And so here we start laying down hydroxyapatite onto the cartilage extracellular matrix. So what we have here is not true bone, but calcified cartilage. In our last zone, the zone of bone deposition, what we actually have is the lacuna walls between the chondrocytes break down, and that leaves these big open spaces. So we have these big open spaces with, which used to be the lacuna with the chondrocytes in them. We've actually broken down the walls between these stacks of lacuna, making big tubes surrounded by osseous, calcified cartilage extracellular matrix. That leaves these long open channels where there used to be stacks of cartilaginous lacunae, and bone depositing osteoblasts will enter into this and the osteoblasts will fill in this area and will then start laying down osteoid tissue, which then becomes calcified into true bone tissue. And that makes trabeculae of spongy bone. So we're making spongy bone inside of here. The compact bone is almost always made by the osteogenic layer of the periosteum. As this cell lengthens, so you can see up here, right, the first bone that gets laid down right there, that's spongy bone. And then as the bone lengthens, that spongy bone will be carved away, at least most of the spongy bone will be carved away by osteoclasts to make the marrow cavity. So this leads to the question of what happens if this goes long, or if this goes wrong. There are actually several forms of dwarfism, but two that I want to talk about right here. achondroplastic dwarfism. A means without, chondro means cartilage. And so with achondroplastic dwarfism, what happens is that you never have your pubescent growth spurt. So your long bones never grow longer. All your other bones, your flat bones, your irregular bones, your short bones, those all grow because those grow through a different method. They aren't growing through the epiphyseal plate or anything like that. So you end up with a normal torso, normal skull, fairly normal pelvis. Your long bones will be of an appropriate width because they're still growing wider, but they don't grow longer. So, and this usually takes place in early to mid-adolescence. So what you end up with is a person who has a normally proportioned trunk and then shortened limbs, because the limbs are the ones that are going to be growing through endochondral ossification. Pituitary dwarfism is actually a lack of growth hormone. So we'll talk about this next term, but growth hormone is going to be the hormone that stimulates cell division and cell growth. If you lack enough growth hormone, if you're making too little growth hormone, then all forms of cell growth will be stunted. So somebody with pituitary dwarfism will have an equal deficit in both the lengthening and withening of the bones. So they will end up smaller because they're doing less growth, but both of their bone growth mechanisms are being stunted, so they end up with normal proportions, just smaller. Of these two, achondroplastic dwarfism is significantly more common than pituitary dwarfism. Our second type of growth is bone widening and thickening. So this is not going to make our long bones longer, but it's going to make all bones wider and thicker. They're basically going to grow equally in all directions. And this is what's called apositional growth. Growth that doesn't occur at a specific position. It's going to occur at any bone surface. And this is actually the way almost all compact bone is made. What happens is that the periosteum layer, so the periosteum has the osteogenic layer on its innermost surface. That osteogenic layer has osteoblasts. And what you do is you just lay down bone tissue, and the bone gets thicker. Now, you don't actually want your bones to just get thicker. They would end up being really, really heavy. So while you are laying bone down on the outside of your bones and continuing to lay down more and more compact bone, the osteoblasts are becoming trapped as they lay down bone tissue inside of them and becoming osteocytes. Then osteoclasts on the inside of the bone start chewing away bone at the bone marrow cavity. And so you're laying down new layers of bone on the outside at the same time that you're eating away layers of bone on the inside. So the bone doesn't actually end up being much thicker. You're just laying down bone and then eating away bone, so it ends up just growing outwards. When you lay down layers of bone from the from the periosteum, you basically lay down a layer of cells, right? So you've got your osteoblasts from the endosteo layer, or the osteogenic layer. They move in, right? So let's say you have so let's say that this is the surface of your bone here, and you have your cells of the endosteum. These are your osteoblasts. Coating it. Well, your osteoblasts lay down bone extracellular matrix around them. And they get trapped inside and become osteocytes. So now you can see they've laid down bone all around them, and we have our osteocytes inside here, and they just become your osteocytes. And then you have another layer of osteoblasts along the outside. These osteoblasts, by the way, come from the osteogenic layer. There are osteogenic cells in the osteogenic layer. They divide, and when they divide, the new cells become osteocytes, or osteoblasts, that then lay down more bone. And basically what you're doing is you're laying down circumferential lamella, right? So each layer of osteoblasts is going to go into making up one of these lamella. And then at the same time, on the inside of the cell here, on the bone here, right? So we've got a whole bunch of like bone tissue all the way through. We're making our bone thicker. So what happens is on the inside, we have our large osteoclasts that look like the little ghosts from Pac-Man. And as we make our bone thicker, they're going to secrete things that will actually dissolve bone. And as we make it thicker from the outside, we make it thinner from the inside. So to recap, what you need to know for this video is the two methods of ossification: endochondral ossification and intramembranous ossification. You need to know the steps of each and also when and where they would be used. Intramembranous is going to be used for like pretty much all the flat bones. Endochondral ossification will be used for the long bones. You need to understand the metaphysis and or epiphyseal plate, kind of the same thing. Know the zones of the metaphysis and what's happening in each of them, and you should be prepared for questions about what might happen if you were to disrupt one or other of the layers. So know the function of each of those zones. You need to understand the process of apositional growth and how that is going to contribute to bone widening. And you need to know those two forms of dwarfism that I discussed, achondroplastic and pituitary dwarfism. Know the differences between them and which system is being disrupted in each.